Hi guys, what's up? कैसे हैं सब लोग? So today we'll be starting with the second chapter of Unit 1, that is biological classification. And again, I'm dividing this chapter into about, I think, 3 to 4 parts. So I'll let you know what we're going to do today. So we're taking very easy topics for today. That is firstly classification and the various kinds of classification, followed by a small overview of Kingdom Monera. That's it, and we'll be starting Protista in the next class. Okay, so we studied the need for classification in the last chapter. That is to make studying organisms more easier, right? So how did people actually classify organisms? What is the entire history of it is what we're going to learn in the first page. So, you know Aristotle, also known as the father of biology. So, in that time, his knowledge was very primitive. So, he classified plants according to their general morphological characters into like herbs and shrubs and trees, जो हम third और fourth class में करते हैं वैसे. And animals he classified by calling them either animals having red blood or having no red blood. All right, and then comes two kingdom classification provided by Linnaeus. And a question for you: do you remember what Linnaeus did? We did this in the last chapter. So, if you don't remember, Linnaeus is the person who discovered the nomenclature for naming organisms, the scientific notation. All right, proceeding. What Linnaeus said was that all organisms can be divided into plantae and animalia. Now you only think there are millions of organisms. We learned that 1.7 to 1.8 million species exist in this world. Do you really think all of them will come under plantae or animalia? There's so many organisms which are unicellular. There are prokaryotic organisms. There are autotrophic, heterotrophic. There are so many characters we can consider in order to classify organisms. So that's why this theory wasn't adequate enough to classify organisms. Now, not only was there two kingdom classification, there was a three kingdom, four kingdom, etc. But at the end, the most acceptable one we have is the five kingdom classification by R.H. Whittaker, and that is that he divided all organisms on this earth into Monera, Protista, fungi, plantae, and animalia. And just check out the table here, it's very important. It shows you the different characteristics of all of these organisms based on their cell type, based on their cell wall, nuclear membrane, body organization, mode of nutrition. If you look at them carefully, there are certain terms you need to note. For example, what is Holozoic? Holozoic nutrition is exhibited in amoeba and also in animals, in which the food is first taken in and then digested. All right, and they've also mentioned some things like the cell wall composition. For Monera, for bacteria, it's made up of peptidoglycan, which is a polysaccharide. All right, and in Protista and in fungi, it's made up of chitin. This is also very frequently asked. And in plantae, we all know it's made up of cellulose. And in animals, cell wall is absent. This we've learned long back. Alright, so just go through this table. Another question you can get is what characters or what factors did R.H. Whittaker consider in order to divide these organisms? And they are given in this line. Alright, this can be a very important one marker. And the characteristics are cell structure, body organization, mode of nutrition, reproduction, and phylogenetic. Okay, what is phylogenetic relationships? It's basically the evolutionary relationships. If you remember, we learned systematics in the previous chapter. It's the same thing. Also, there is a three-domain system. In the previous chapter, I had left out one of the hierarchical taxa. But if you remember, dumb kids prefer cheese over fried spinach, the mnemonic used for remembering the hierarchy. So in that, the dumb referred to domain. Now, domain is something which is higher than kingdom. And there was a three-domain classification which was proposed by Carl Woese. Okay, they haven't given much about it in NCERT, but all you have to know is that in the three domain classification, he just divided all these groups into three domains. That is, Monera is being divided into two other kingdoms, that is Archaea and Eubacteria, which we'll be learning about further. But what you have to remember is there are three domains and six kingdoms in this particular classification. That's it. Alright, if we proceed and look at the next paragraph, ये ज़्यादा कुछ नहीं बोल रहा है. It just says that जब हम characteristics बदलते हैं, that means when we change the basis of classification, then the organisms included in one group can also shift to other groups. For example, if you take Linnaeus' classification, animals and plants, his classification included bacteria, mosses, plants and angiosperms, gymnosperms, everything under plant. But you all know that these are different organisms, right? They all of them were included under Plantae only because all of them had a cell wall. All right, but we know that bacteria are prokaryotes and the rest are eukaryotes. So when we take prokaryotic and eukaryotic as the basis, then we can classify these into different groups. So similarly, they've given a lot of examples. For example, Chlamydomonas and Spirogyra. Chlamydomonas is a unicellular organism, all right, and Spirogyra is multicellular, but it's still placed under algae together. This is because they have the same thallus structure. We'll learn what a thallus is in the further classes, all right? So yeah, and there are a few more examples given. You just have to read this paragraph. There's nothing much. And we can begin with Kingdom Monera right now. Kingdom Monera includes only bacteria from all walks of life, okay? And it only includes unicellular prokaryotic organisms, all right? Remember, unicellular prokaryotic. Ab dekho yaar, bacteria is everywhere. Okay, they can be in soil, they can be in extreme climates, they can withstand any kind of climatic condition, because they're that diverse and they have such properties in their cell wall, all right? And they can be found as parasites on organisms, they can be found in snow, they can be found in hot water springs, everywhere, all right? And then we move to the classification of bacteria based on its shape, okay? There are four basic shapes that a bacteria can possess. First one is cocci or coccus if it's singular. So cocci is spherical shaped, followed by rod shaped bacillus, and then spiral shaped spirulina, and comma shaped vibrium. Okay, comma shaped. If you just look at these, so these diagrams are important. It's important that you remember the shapes and its associated name. Okay. And they've also mentioned that bacteria is the most diverse organism metabolically, because they can either be heterotrophic, autotrophic, chemoautotrophic. Okay, what is chemoautotrophic now? You know photoautotrophic organism, that is photosynthetic organisms, right? They utilize sun's energy in order to make food. Similarly, they utilize these chemosynthetic organisms. They utilize the energy from chemical reactions to get their nutrition. Okay, and heterotrophs depend on other organisms for their own food, just like humans. Now, there are two groups of bacteria which we will be studying here, Archaebacteria and Eubacteria. And if you remember the three domain classification, then the first domain consists of Archaebacteria and the second domain consists of Eubacteria. Okay, Archaebacteria are those bacteria which can live in any kind of climate. Okay, I mentioned before also, and they've given a few examples here. The bacteria that can live in extremely salty areas, you know, sometimes the seawater is very salty, such bacteria are known as halophiles, and halophiles always refers to loving. That is, if you call anything hydrophile, then it is attracted towards water. Similarly, halophiles are those organisms which can survive in salty water, salty areas. Hot springs, I told you they can live in extreme temperatures. So hot springs are known as thermoacidophiles, and certain bacteria can also live in marshy areas. These are known as methanogens, okay? And they have a certain kind of cell wall which helps them survive in these conditions. So this cell structure and the proteins produced by it help it survive. Okay, and methanogens. Okay, this part comes in your 12th grade too, but it's necessary that you remember this. Methanogens are present in the alimentary canal of certain ruminant animals. Ruminant animals refer to animals that feed on cellulosic substances which can digest cellulose, like cows. Okay, so cows and buffaloes, when they digest cellulose, these methanogens, they are the bacteria that help in the digestion of cellulose, and they grow on cellulosic substances. So when you get the fecal matter of these ruminant animals, you can find these bacteria, and these bacteria are again you are made to undergo a process by which you can produce biogas, which is a very sustainable way of energy production. Okay, you'll be learning about that very soon. Now, the second group of bacteria we'll be learning about is eubacteria. Eubacteria are also known as true bacteria. Okay, they have a cell wall and they might or might not have a flagellum. Okay, cyanobacteria, this name, you'll be coming across this name in many chapters in your 11th and 12th biology. So these are also known as blue-green algae, and they are photosynthetic bacteria. Okay, photosynthetic autotrophs. They synthesize their food by utilizing the energy of the sun, and they have the pigment in them is chlorophyll A. If you remember, plants are able to photosynthesize because of the presence of chlorophyll. Now, chlorophyll has different types: chlorophyll A, chlorophyll B, and several other pigments. So, chlorophyll A is the pigment in cyanobacteria. Okay, cyanobacteria can be filamentous, colonial. Colonial is a lot of cyanobacteria in a given colony. You know, colonies of bacteria. You've heard about it many times. All right. And they can be freshwater, they can survive in freshwater, they can be marine, they can survive in seawater, or they can also be terrestrial. Okay. Now, since they form colonies, these colonies usually have a gelatinous sheath on them. Gelatinous sheath is, it's basically a mucilaginous sheath which protects it from environmental perturbations. Okay, and okay, this point, they often form blooms in polluted water body. What is a bloom? Bloom, if you notice some lakes or rivers which haven't been used for a long time, they develop this green layer over them, right? This very dirty-ish layer. So that layer is made up of mostly algae and this cyanobacteria. Okay, so they form blooms on water bodies, and some of these cyanobacteria also help in fixing nitrogen. If you remember nitrogen fixation from your previous classes, you know that there are certain bacteria in the soil which help in converting atmospheric nitrogen into ammonia, right? So that it provides plants with nutrition. So these nitrogen fixing bacteria are also a part of cyanobacteria, and they have special cells or structures known as heterocyst. You see this diagram they have given in NCRT. They've also shown the mucilaginous sheath I was talking about. Okay, so basically they have this heterocyst, and this particular cell is the one which helps in nitrogen fixation. Okay, and examples for it are nostoc and annabella. This example is pretty important, so learn them. And chemosynthetic autotrophic bacteria. I've already told you what chemosynthesis means. So what these bacteria do is they oxidize various inorganic substances such as nitrates, nitrites, and ammonia. And when they oxidize this, a certain amount of energy is released, and they utilize this energy as a form of their nutrition. Okay, and nutrition is required for ATP synthesis. So they utilize this energy for synthesizing ATP, and ATP, as you know, is the energy currency in a cell. Okay, And they also play a great role in recycling nutrients because obviously they're oxidizing nutrients, so they help in their recycling too. Okay, now we're done with photosynthetic bacteria, chemosynthetic bacteria. We're coming to heterotrophic bacteria. All right. Now, heterotrophic bacteria, they depend on other organisms for their nutrition, right? And they have many functions, they occur in many forms, okay? And some of them are very important decomposers. Now, decomposers are those organisms in the soil which convert complex organic substances into simpler inorganic substances. And you will be learning the process of decomposition in a chapter called microbes in 12th grade. Okay, and there are several other functions they have. They help in curdling of milk when you add lab bacteria, that is lactobacillus bacteria, to milk. They help in breaking down milk protein and they convert it into curd. They help in nitrogen fixation. If you remember rhizobium bacteria, it fixes nitrogen in leguminous plants, right? And some of them are pathogens. Pathogens are basically those organisms which cause diseases in humans or plants, etc. And some of the diseases caused by them are given here. That is cholera, typhoid, tetanus, and citrus canker are diseases caused by bacteria. So you might get a question in NEET saying which of the following diseases are caused by bacteria or which of the following are caused by viruses. So at that time, you'll have to remember these diseases, all right? And bacteria, they mainly reproduce by binary fission. Binary fission is when a single cell divides into two different cells and the nucleus also splits into two daughter nuclei. But sometimes they can also undergo sexual reproduction, okay? Because these bacteria, they have this circular DNA known as plasmid DNA, okay? So this plasmid DNA helps in sexual reproduction of bacteria under unfavorable circumstances, okay? And this example of mycoplasma is a very frequently asked question in your theory paper. Mycoplasma does not have a cell wall and it's one of the smallest living cells which is known, and it can survive without oxygen. That is, it's a facultative anaerobe, okay? And mycoplasma is also a very well known pathogen. It causes many diseases in animals and plants. And also, it's very important for you guys to see each and every diagram in NCERT because they'll ask you a lot of questions from diagram. Diagram based questions is where people usually tend to lose marks because they ignore the diagrams. So you'll have to learn all the diagrams along with their labeling, each and every labeling, if you are aiming to score full in any of your paper. So we're done with Kingdom Monera. In the next class, we'll start with Protista, and if time permits, we'll also start with fungi. Alright guys, thank you so much for listening, and if you have any more suggestions, if you have any questions, then please write it down in the comment section. And you can also tell me which unit I should proceed with next because I don't really have any particular order. It depends on which one you want to study. I'm fine with anything. And yeah, please like, share, and subscribe and send it to all your friends who might need it. Thank you.